Well, secondary active transport is a challenging concept to get. It's not made any easier by the lame name, secondary active transport. Obviously, primary active transport uses ATP to make a concentration gradient. This one does not use ATP. It uses that concentration gradient from the primary active transport to move something else against its concentration gradient. See, not an easy concept. Well, let's explore it a little bit further here. Well, here's maybe a bit of an analogy for it. Let's say I have a note. I want to pass it to somebody across the room, but I really don't feel like getting up and walking over there. I mean, that would be more like primary active transport, right? So instead, I'm going to put energy into another system to propel the note to the other side. Here you go. So that is kind of like primary active transport. I'm directly spending energy, or ATP, to blow up something and create a concentration gradient. Well, this one's a pressure gradient. There's pressure inside the balloon, right? So now I can just stick this note on. Well, that was not very effective. It works better for the cell. Let's take a look. In order for secondary active transport to work, we need to have a concentration gradient. It can be sodium, hydrogen, variety of other ions, but always something like that. What made them concentrated? It was probably the sodium-potassium pump. And remember, it uses ATP from the cell to pump sodium out. 3 sodiums out for every 2 potassiums in, but all we really care about here is the sodium. So now we have a concentration gradient. Sodium wants to move into the cell. In a way, this is kind of like facilitated diffusion of sodium into the cell with a hitchhiker, glucose. But let's call it by the more proper name, secondary active transport. Here's how it works. We have this giant carrier protein. It needs some power to move glucose against its concentration gradient, and that power is going to come from sodium. So they all find sort of their places. They're dumped into the cell, and then that resets. And as long as you have a concentration of sodium that wants to come in, you can bring more glucose along with it. But that's basically how it works. You know, a lot of the discoveries about cells that we talk about in this class are really pretty old, but this one, not so much, surprisingly enough. And it plays a big role in the digestive system, as first described by Robert Crane in 1960, as playing that role in the digestive system. I mean, this was after DNA was described, so pretty recent. Let's take a look. Here's our small intestine, where we absorb all of our food. And so here's that hollow inside, the lumen, and then around that are lots of blood vessels, and we're trying to get the food from the lumen into the blood so it can go around the body and nourish us. All right, so taking a closer look at that, here is the lumen, this space where all the sodium and glucose is. Now, you eat food that contains starches and other sugars, and so you've broken those down into glucose. But we need to get that into the cell. The idea is we're going to pack this cell full of glucose. That's why I have lots of glucose drawn in here, so that it can leave by facilitated diffusion into the bloodstream. It has kind of a modest amount of glucose in it. There you go. But to get facilitated diffusion to work, I need to get a lot of glucose into here. That's where this protein comes in. So this is a co-transport protein that's going to use secondary active transport. So it's going to round up a couple of sodium and a glucose, and they're all going to come in together. So sodium's moving down its concentration gradient. The cell makes sure it's empty of sodium, and then glucose is moving against its gradient, but since it's powered by sodium, it still comes in. This is important for the absorption of a lot of nutrients, including glucose and many amino acids, even the nucleotides from the broken down DNA in our food. But it doesn't stop there. It is involved in our kidneys as well. So when our blood spills out sodium and glucose into the urine, the kidney brings those both back into the bloodstream because we want to hang on to both of those. They're important aspects of our blood. Otherwise, we would eat food and then all the salts and sugars would leave through our urine. It would do us no good. But these transporters are in all kinds of cells throughout the body, helping to bring glucose and amino acids and other important biomolecules into the cells, usually with the help of some sort of ion. There are other examples as well where one ion is exchanged for another one, but I'll leave that for a future anatomy and physiology class to describe. Well, there we have secondary active transport. Shows us that a concentration of ions actually possesses power to do things like move other ions or biomolecules against a concentration gradient. Plays an important role in a variety of systems. Well, that's it for this time. Until next time, keep appreciating life, even the confusing and little things like secondary active transport.